Okay, so next we have a SCORP team, or veterans included, sorry. But I noticed that these are 15-minute slots, so they're kind of related, I think, a little bit too. So this whole theme this afternoon is about bioassessment. So next we have Betty Fetscher, who used to work at SCORP. She's now at San Diego Regional Water Board, swamp coordinator, and your degree is from another UC, not Davis, right? UC San Diego. Okay, so thank you, Betty, for coming and speaking.
Sure. So there are many faces to algae, and I wanted to sort of go over some of the faces of algae because I think it provides a nice context for me to sort of talk to you about how there are algal connections to a lot of different state programs, not the least of which is bioassessment, but there are also other connections that are interesting. So algae is my friend, and algae should be considered your friend. Why? Because algae provides a lot of valuable services, not the least of which it helps you breathe, but also algae assimilate carbon and nitrogen into aquatic food webs, which benefits bug communities and in turn fish communities. And as Andy touched upon, and I'll touch upon a little more, algae are actually really good water quality indicators. So algae are great, except for the fact that algae are also horrible. So one of the things algae does is if it's overstimulated with nutrients, it starts to grow like crazy and it creates algal nuisance. So it can be unsightly, it can cause weird blockages, it can interfere with fish habitat. When it dies, it can suck the oxygen out of aquatic systems, it can smell terrible, it can cause taste and odor problems to drinking water, and some algae, blue-green algae or cyanobacteria, can produce toxins. So there are some good beneficial uses associated with algae, both as a contributor to beneficial uses itself and as a monitor of beneficial uses. But there are also some things that we need to sort of worry about what algae does, and some of our regulatory actions are to try to control algal nuisance. So a little bit of history of algae and bioassessment in California. Algae have actually been around for a while. They were monitored as part of the California Monitoring and Assessment Program way back when. Back then, we didn't have ways of actually interpreting the algae data that were specifically tailored for California. And so different regions in the state found some small sources of money that they were able to use to sort of create regional indices of biotic integrity. So Andy already talked to you a little bit about one of the algae IBIs. So there was some early work in the Lahontan region by Dean Blinn and Dave Herbst, and then later Region 3 and Southern California both developed indices using Prop 50 money. And by this time, the swamp statewide programs, perennial stream assessment, reference condition management program, and some regional programs, the Southern California Stormwater Monitoring Coalition and the Bay Area Regional Monitoring Coalition started collecting algae data. And we now have well over 1,500 sites worth of algae data, which is great. We can start doing algae-based assessments. And we also have a lot of data to continue improving algae biomonitoring tools. So some places only look at diatoms. Diatoms are single-celled algae. They're enclosed in these little glass pill box-like structures. There's an example of a diatom scum. Smells like fish because fish smell like diatoms. There's some live diatoms here in the middle. This thing doesn't seem to work very well. And then there's a nice cleaned-up diatom on the right. We don't only look at diatoms in California, though. We also look at non-diatom or soft algae. Went out on a little bit of a limb, including this. Thank you. Well, I need a clicker too. Because a lot of places don't use soft algae, but it turns out that they provide us a lot of valuable information. So that ended up being a lucky stroke. And within the soft algae, we also include, again, cyanobacteria. So really quickly, because some of you might have seen algae data coming your way, but you're like, what is this? I just wanted to talk really briefly about how samples are processed and what kind of data you get out of it. So with diatoms, basically you boil them in acid so that you destroy all the organic matter, and what's left behind after you mount them and look at them under the scope is just the plain old glass-like structures that the diatoms have as their shells. The soft algae, it's a little bit more complicated. Oh, and so you count 600 of those. We call them valves, the little lids on the diatom. Soft algae is a very different animal. Generally, it's analyzed by a different lab. There's sometimes big, like macroalgae. There's sometimes small unicellular microalgae. The big stuff is the volume of it is measured by displacement, and it's analyzed sometimes under a dissecting scope. The micro stuff is analyzed under a compound scope, and basically the lab counts 300 entities of micro and macro combined. And then there's some other stuff that's looked at, but a little bit too esoteric for today. So now I just want to talk, I'm kind of going to be all over the board for the end of this presentation. I just wanted to talk about some different ways that algae are involved in some of the statewide programs, either right now or anticipated to be. So for one thing, and Andy has already touched on this quite a bit, algae are used in bioassessment, and we're finding in Andy, all I think about is algae. Andy helped us understand that algae actually complement bugs really well in bioassessment. It's a team. Algae and bugs are a team in bioassessment. It's the old 1-2 punch. So they have high dispersal rates. They have high reproductive rates. They can respond in changes to water quality, whether there are degradations or improvements, very, very rapidly, probably on a shorter time scale than bugs. So there's a temporal complementarity going on. As Andy noted, they're highly responsive to water quality. And in fact, to some degree, unlike bugs, they're really not very constrained by available habitat. They're mostly focusing on the water, less bothered by the habitat. I'll show you a data slide on this. But there's also interest in algae beyond bioassessment. So for one thing, and Jay Davis talked about this a little bit, algae can produce toxins. So I'm going to talk to you a little bit about some of the data we have specifically for streams in that regard. And then finally, algae will have some kind of a role in the development of California's nutrient policy. So let's take a quick look at what that might be. So in the next few slides, I'm going to talk about each of these three topics. So first, I want to talk about sort of the habitat, the fact that algae are somewhat immune to changes in their habitat. So here are these 4 different panels correspond to bugs, soft algae diatoms, and then the hybrid index. This is the H20 that Andy was talking about. And on the x-axis is the index scores for each one of these assemblages, and higher is better. And on the y-axis, this is density. So these are basically like smoothed histograms is the way to think about them. And the pink is concrete. So the pink corresponds to sites that had concrete lined channels. And the blue corresponds to all other types of sites, regardless of quality. They just don't have concrete. And so what you see in the bugs is they max out fairly well below their full potential of scores that they could achieve. But when you look at the algae, at least some portion of the population of concrete streams is achieving very high scores, and it really is gangbusters in the diatoms. So what you're seeing is algae are not really necessarily held down by bad habitat if the water quality going through the system and being sent downstream is high quality. So now let's talk really briefly about toxins. So Jay brought up the story about the sea otters in Monterey Bay that died from liver failure caused by consuming microcystins that were derived from inland sources and came down to the bay through tributaries. So after a lot of bioassessment had been going on throughout the state, I was a little bit curious about, cyanobacteria. Do we have a lot of benthic cyanobacteria in our streams? Surely we do. So this map shows basically the yellow dots are sites that have, where we recorded at least at genus level, benthic cyanobacteria that are known from the literature to be able to produce toxins. The red sites are ones where at the species level we know that they can produce toxins. And the blues are sites where we did bioassessment, but no cyanobacteria that could produce toxins, known to science anyway, were recorded. So well over 90% of the bioassessment sites that have been visited to date across the state had benthic cyanobacteria that's capable of producing toxins. But are they? So we actually were lucky enough to get all these different monitoring programs to collect an extra sample that we analyzed for cyanotoxins. We focused on microcystins. However, we did look at anatoxin and saxitoxin and some others at a more limited scale. We found that 33% of our sites were producing microcystins. The bigger the dots and the pinker the dots, the higher the concentrations. One of our first questions was, is anthropogenic activity exacerbating toxin production in these systems? So far, we don't have any evidence to that effect. In fact, some of the highest levels of microcystin that we found were in relatively undeveloped sites. So right now, unlike with lakes and reservoirs where planktonic blooms, there's substantial evidence that shows that anthropogenic activities are increasing the threat of toxic events. So far, this is basically just a baseline. We don't know where this is headed or necessarily what it might mean. But one thing's for sure, I mean, if a toxin like microcystin that has a long half-life on the order of 10 weeks is being produced in these streams, it definitely has the potential to get exported downstream where he and others live. So finally, I wanted to talk about sort of the connection with nutrient policy. So in order to talk about that, I just wanted to mention, we already know from Andy's talk, algae respond highly to water quality. They really respond to nutrients. So in these two graphs, on the left, I have total nitrogen concentration along the x-axis. On the right, I have total phosphorus concentration. On the y-axis, and this is for the diatom community, we have NMS ordination. It's basically a way of summarizing community data, squeezing it down into a single value. So as you move up and down on this axis, you're seeing sort of a quantitative way of assessing changes in community composition. So first off, we see a very, very, very tight relationship between algal diatom community composition along a nutrient gradient. But, so the community is changing as you crank up the nutrients. My collaborators at Cal State San Marcos a few years back spearheaded a study where they looked at activation of genes that are responsible for fixing nitrogen, nitrogenase genes. So they were looking specifically for nitrogenase production in the small subset of diatoms that actually are capable of fixing nitrogen because they contain little organs that are vestigial blue-green algae that still maintain their nitrogen fixing capacity. And what they found is nitrogen fixation genes are turned on all along this gradient up until roughly about here. And then they turn it off because it's really expensive to fix nitrogen. And you don't want to turn that on unless you need it. Well, you don't need it as you start getting more dissolved inorganic nitrogen in the system. So just as nitrogen fixation starts to wink out, you start seeing major changes in the community. The nitrogen fixers start disappearing because they're no longer competitive. So that's all by way of saying that algae are really, really well tuned to giving us information about stream nutrient status. So I'm going to wrap it up here just talking about some of these sort of ongoing initiatives that are occurring right now in statewide algae program. It so happens that SQURP is taking the lead on all of these initiatives, partially supported by SWAMP. So one is helping out with doing the science that's going to support the statewide nutrient policy. And one of the things they're considering right now is using algal community composition, not just the traditional algal biomass. And that's something that they're going to use a biological condition gradient to help with that work, and Tetra Tech is going to be involved. The next is, Andy mentioned that CSCI, remember that bug index is a modeled index. So the idea is to do the same kind of thing for algae so that there's a statewide modeled index. SQURP's going to be leading that effort. They're also going to be working on a standard taxonomic effort in collaboration with folks at Cal State San Marcos. And finally, there's a lot of reasons that are probably too involved to go into, but we're seeing a lot of reasons why we should be working on developing molecular tools for analyzing algal data for use in bioassessment, rather than forever relying on the current morphological taxonomic-based data. So that's another effort that's getting kicked off. With that, just wanted to acknowledge all of the different groups that have been involved in helping build algal bioassessment and otherwise infrastructure within the state. And I don't know, do I have time for questions? Okay.
So I got a two-part questions. One, first part is is there a certain criteria for, there are more than 60% of streams are intermittent in the state. And then what is the criteria or the standard procedures to incorporate those systems? I guess we would say in a way as in sort of, is there a sort of an index period for both algae and macroinvertebrates? Second part is the so differences about the substrate, because my experience has been that when I was using, say, standard the sort of tiles versus natural substrate, there has been some differences in the communities, and then also, as you said, it's algae respond to the water changes or water availability pretty quickly. So, what time frame are you looking at the algae index or algae
That's
community?
3. That's 3.
I can go on if you want.
So for the question about dry, there are various groups right now. One of them is in my region who are actually looking at bioassessment, sort of testing the limits of how short-lived a stream can be, an intermittent stream, right, a non-perennial stream, how short-lived that can be, and these indices still work well. And so the bottom line is so far bugs and soft algae are working very well, but kind of shocking everybody, even in the very short-lived streams. Diatoms, for some reason that we can't fully comprehend, are the ones that don't look like they're doing quite as well. Some of the metrics look like they do great, but not all of them. And so the index itself is not performing as well as we would have hoped. I would have actually put my money on diatoms out of the 3. So we're still kind of scratching our heads over that one. So the second question was the substrate, the tiles versus other. So I would agree that certain taxa will have affinities for certain substratum types, but the indices that we're using apparently are fairly blind to that. So the indices are based upon sort of water quality type responses. And so there could be a swap out, like this taxon might do great on a tile, this taxon might do great on sand, but either one of them would tell you the same thing about water quality when it's rolled up into this summary form of the index. And then the third question, I don't remember the third question. I even had an answer, but I don't remember what the question was. Oh, yeah, time response. So I have a couple responses to that. I read a paper once where they put it at 3 weeks. They basically did substratum transplant experiments, and they found complete turnover in the communities in less than 3 weeks. And the other thing that I would respond to that is I've gone to streams that were dry, like bone dry, had a storm, and I visited the stream 10 days later, and I was already seeing blooms. So they can kind of do their thing pretty quick.
Standardized timeline to do sort of a cross-line assessment in a way.
Yeah, so if you want to do bugs plus algae plus everything, yeah, I mean, there are index periods. Those are built into the standard operating procedures that SWAMP sort of puts out. And so right now, we still sort of hang our hat on this notion of these tools are intended for perennial streams, but we're finding that they do quite well, even in non-perennial, and I would imagine that in the next generation of the SOP, or maybe there might even be interim guidance to that effect. I think somebody was over here.
Sorry. You indicated that the half-life of microcystin is 10 weeks. How did you determine that?
I didn't. I got it from the literature.
Yeah, because they have indicated that it depends on exposure to UV radiation.
Yeah.
If there's too much UV radiation, it degrades faster. So I wanted to know how you arrived at the 10 weeks.
Yeah, so the paper that I can't provide you a citation off the cuff. I have it in a paper I published, but this was supposedly under ambient conditions. The 10 weeks was the half-life under ambient. So not like closely, carefully pampered lab. I'm not sure exactly how it was done, but